Hey everyone, thank you for having me here. I'm Wenhuan Zeng, and today I'm going to present our recent studies named NanoGraph, which primarily introduce a framework that classifies nanopore sequencing reads directly from its raw signals using deep learning approaches. Before we dive deeply into this framework, I'd like to give some introduction to the background. So as we know, nanopore sequencing is one of the techniques for performing DNA sequencing, and DNA sequencing is fundamental to understanding the function of genes and other parts of the genome. Because of the significance, sequencing technologies have been continuously evolving since the 1970s. Over time, we have seen a remarkable transition in sequencing methods from the first generation to the third, each with their own characteristics. And the first and second generation of sequencing are represented by the Sanger sequencing and Illumina technology. The major difference between this is the sequencing volume. And more recently, the third generation sequencing technologies like Oxford Nanopore and PacBio have emerged. They overcome limitations of previous technologies by generating longer read lengths. Nowadays, the long-read sequencing getting increasingly popular as it has advantages across multiple aspects. In long-read sequencing, DNA fragments do not have to be broken into smaller fragments for sequencing and then assemble them back and for data analysis. Long and ultra-long reads are more likely to span entire regions of repetitive sequence and structure variation, offering a more complete view of genetic variation and the possibility to reconstruct complex genomes. Especially nanopore sequencing also offers the real-time analysis in time-saving manner. Oxford nanopore sequencing devices rely on flow cells for sequencing to take place. This where the nanopores are host. When a molecule passes through a nanopore, the current is disrupted to produce a characteristic squiggle. The squiggle is then decoded using base calling algorithms to determine the DNA or RNA sequence in real time. And once we obtain the DNA sequences, we can perform a series of downstream analysis of our interest. And here is one of the possible application scenario. And here we roughly summarize the workflow of exploring the composition or contamination of samples. And here we can find that using nanopore sequencing technique, it's unlike the previous technologies. The nanopore sequencer will first generate the raw signals and then performs base calling to produce the sequence of bases for further processing. Although this happens in real time, these steps are also tends to be computationally demanding and memory consuming. And this characteristic has led us to wonder, can we apply deep learning methods to simplify the taxonomic assignment procedure? Driven by these questions, we have developed NanoGraph, which allows us to get taxonomic assignment directly on raw signals in a time-saving manner. And here is a NanoGraph workflow. And firstly, we trained NanoGraph using simulated squiggles, as we are in collaborations with a wet lab which focuses on bioreactor topics. We collected reference genome from NCBI RevSeq across 5 organisms of their interest. And then we simulated the squiggles using Deep Simulator and then map the squiggles to graph structure for further model training. And the figure B shows how we convert the squiggles into a graph. So for each raw signal, we that duplicated the signal values and mapped them to the nodes in the graph. And the neighboring values in the original row signal are connected by weighted edges, and the weight is the variance between the neighbor values. So by adopting this graph structure, our signal can in different lengths, and due to the characteristic of the topological graph, it can capture complete information carried by long and ultra-long read lengths. And the processed graph is then used to train the model, which is built by the graph attention neural network. Additionally, we fine-tune the model on real squiggles provided by our collaborators using transfer learning to enhance the ability and performance of our models. In the graph structure, so to demonstrate the effectiveness of graph data structure, we have visualized the representations across different types of input. So here we can see the figure A. So the sub-figures located at the upper part of the figure A were generated by the numeric inputs with different lengths, and the figures located at lower part are generated by the graph embeddings, which encoded by the graph attention neural network, and the bottom left and bottom right is different from the number of the head in the encoder of the encoder. And here we can find that the graph embeddings can better capture the characteristics of samples, as they show improved differentiation between categories compared to the numeric input. And then let's move on to the right side of the slides. The figure B and C display the model's performance using the confusion matrix, and they are generated by the models that trained on the simulated and the real signals respectively. And the model trained on the 5 organisms can correctly classify most of the samples except for the samples from the 2 species, which these 2 species have quite close relationship as they from the same genus and in terms of taxonomic lineages. And for the other species, we assume they performs better because they are not, for the top 2, they just share the same order instead of the genus. And after we use the transfer learning, the model trained using limited number of real signals also shows some abilities to classify the samples for now. And additionally, to have a more complete comparison of our methods, we also benchmark it with the previous study across the different combinations of organisms. And here we can find that compared to the previous study, our models outperforms across all the combinations. And the data set is more well processed as they have the fixed length. So I think the performance is better than the one we applied to the generated squiggles. So to enhance our model comparison, we perform taxonomic profiling for real signals, which provided by our collaborators using traditional methods. First, base calling the raw signals using Dorado, and when performed bioinformatics analysis using the other tools. And we have visualized the classification results using the Corona and the confusion matrix, and compares the confusion matrix to the previous one. Here, so far the results generated by the traditional methods on the real squiggles are better than our performance for now. But for this traditional method, it will also have some cases that are not able to classify to the other, to one of the species. To recap, we introduces a new data processing methods which can capture the long and ultra long row signals to the topological graph structure. And NanoGraph is a graph-based deep learning approaches that aims at classifying the nanopore row signals to limited species. And there's still room for our work to improve regarding the performance, especially on the row signals classification. And in future, we aim at advancing the model's ability in terms of the real-time manner. Thank you for here. I would like to thank you to my collaborators and group members, and thank you for your attention.